And why I'm interested in these systems. Basically, I'm interested in all things that are mucosal, all surfaces that are mucosal in our body, and this includes lots of examples such as oromucosal, nasal, oral, ocular, and vaginal, rectal, and intravesical. So all these surfaces are mucosal, and they have specific features, but they also have lots of things in common. And the drugs could be delivered via these surfaces. But there's certain tricks you need to learn in order to deliver them efficiently. And one of the tricks is the use of so-called mucoadhesive systems. So the systems that are able to stick to these mucosal surfaces, and they're able to retain on their surface for a prolonged period of time. The first observations, first report on the use of polymers in this area was published in 1947 by Scrivener and Schanz, and they've used a combination of polymers to administer penicillin to oral mucosa. But unfortunately for them, they published it in a dental journal, Journal of American Dental Association. So this paper was practically went unnoticed for many of the pharmaceutical scientists. And the pioneer in this area is Professor Tsuneji Nagai from Japan, who published a series of papers in the early 80s demonstrating that water-soluble polymers, when they are formulated as various formulations as gels, tablets, they could be mucoadhesive and they could deliver drugs efficiently to mucosal surfaces. And if we look at the statistics on publications, you will see that there's a growth in publications in this area. Well, as of a few days ago, I counted 5,243 papers. And where we could see the implication of this interest in the research in mucoadhesive polymers, there are several products on the market, and the number of products are growing. So these include mucoadhesive tablets that you could put into your mouth. They will stick to your gums, so to the cheek, and they could stay in there for several hours to provide extended release. Then there's oral films, there's various eye formulations such as drops and gels. There's some cough syrups that use mucoadhesive polymers to administer certain drugs, and also some oral systems. For example, Gaviscon has alginate, and alginate is mucoadhesive. And when I came to this field, it was roughly in 2005 when I became an independent lecturer at Reading. I started conducting research on various mucoadhesive polymers, and initially we've been working with so-called conventional mucoadhesive materials of first generation. So these are all water-soluble polymers, and they're listed in this slide. I also listed some selected publications. So what we have done with these polymers, we have used them to formulate various products such as tablets, various liquid systems, in situ gelling systems, and also films for ocular drug delivery. But then at roughly that time, there was a pioneering research by Professor Bernkop-Schnurch from University of Innsbruck in Austria. And he identified the need for making the polymers more adhesive. And he pioneered so-called thiolated polymers when he took various water-soluble polymers, modified them by functionalizing them with thiol groups. And these polymers were found to be more sticky to mucosal surfaces compared to conventional water-soluble polymers. And this enhanced stickiness was found to be because of the disulfide linkages formed between the biological surface and the polymer. And Professor Bernkop-Schnurch proposed his very simple method of modifying the polymers. So his group took conventional water-soluble polymers, then conjugated them with thiol-bearing small molecules, and that's the polymeric dimer. They've published around 200 papers on this topic, launched the company, and very recently there was one product launched on the market, so it came through all the clinical trials, and now the product is called Lacrimera, which is thiolated chitosan that is used in ocular field. So when I came to this field, I kind of wanted to have something of my own with enhanced adhesiveness, and my group has proposed a bottom-up approach. We didn't start using conventional polymers to modify them, but we decided to go from other way around. We decided to start from small molecules with adhesive groups, and we converted them either into soluble polymers or some colloidal particles with these adhesive groups. And one of the first attempts to do that was to play with thiolated silica nanoparticles. So we pioneered the synthesis of these particles. There's a number of thiolated silica papers that were before our study, but we changed the synthesis conditions slightly, and we managed to hit into the area where the particles are formed of particularly small size and high yields. So we did a lot of work on these particles and demonstrated that they are highly mucoadhesive. Also, these particles were commercialized by PolysciTech, but unfortunately, these particles were not good model. They were a good model for drug delivery, but they were not good vehicle for drug delivery because they were not porous. We are still struggling to make them porous. And then we also tried some other ways of making particles using bottom-up approach. And we've tried to use some slightly more complicated polymer chemistry. I'm saying slightly more complicated chemistry because it is from the viewpoint of pharmaceutical researchers who do like the simple one-stage reactions. So in this piece of work, what we have done, we designed a monomer with protected thiol groups. We converted it into microgel particles using conventional emulsional free radical polymerization. Then we deprotected the thiol groups and prepared the thiolated microgels. So of course, we have done all the extensive characterization of the systems, and we also demonstrated that the systems adhere to mucosal tissues. How we test adhesiveness? We have developed several methods of our own and also used well-known literature methods. One of the methods is called flow-through fluorescent assay. When we use a piece of mucosal tissue, typically taken from an animal, for example, it could be bovine eye, we put it on a slide and we put our mucoadhesive formulation loaded with fluorescent dye, and then we wash it off with the artificial body fluid. If it is the ocular delivery we're interested, then we wash it with artificial tear fluid. If it is the urinary bladder, for example, then we wash it with artificial urine. And then each wash we analyze by taking fluorescent images and using image analysis. And the graph on the right shows that the microgels with 80% of thiolated monomer, it gave us massively higher adhesion, massively higher retention compared to the particles that have lower levels of thiols or the particles that don't have thiols. So that was one of the systems we developed. But then I also was looking at the literature at the time, and I came across studies by Professor Bernkop-Schnurch from Israel, who demonstrated that if you take a polymer, you functionalize it with acrylated groups or acryloyl groups, then these polymers become also reactive with mucosal surfaces via Michael addition reactions, and this could happen under physiological conditions. I really like this idea because I knew that thiols, they are very prone to oxidation. So there were lots of problems with thiolated systems. For example, they sometimes result in insoluble products. So I took this idea on board and we decided to give it a try using slightly different chemistry. So we prepared nanogel particles from bipolymerization of 2-dimethyl aminoethyl methacrylate. And then we functionalize them by reacting with acrylyl chloride. And we have conducted a series of studies, a series of physical chemical studies to characterize the particles. And we also looked at retention of these systems on ocular surfaces. So the graph at the bottom shows how they are retained on the surface. So if we have the quaternized particles, or how we call them quaternized, the particles that were modified with acrylyl chloride, then we could improve mucoadhesiveness much significantly. Then, because I already said that pharmaceutical scientists, they like easy systems, so we tried to convert this the same chemistry into an easy system. We took a conventional copolymer, which is a pharmaceutical copolymer produced by Evonik, and they're called Udrigid. They have the same groups as in the nanogels before, and we decided to try this reaction to modify it by attaching the acrylyl chloride. Again, the same story, we have improved mucoadhesiveness, but we also started looking at toxicological assays to characterize the irritation properties of these polymers, because mucosal surfaces, they're quite gentle, so you could irritate them. We used quite unconventional assay, which is called slug mucosal irritation assay. When you use live slugs, you put them on a surface containing your materials, and then you measure the amount of mucus produced. If they produced a lot of mucus, then it means the system will become irritant. If they don't, or if they produce the low levels of mucus comparable with our controls, then it means that the system is not irritant. And what we have demonstrated that our acrylated udrigids, they are non-irritant materials. We also have developed a number of methacrylated systems, such as methacrylated chitosan by reacting chitosan with methacrylic anhydride. And we have looked at the model of urinary bladder in this case, demonstrated that the urinary, this methacrylated chitosan is much more adhesive compared to conventional chitosan. And this time, in terms of toxicology, we used conventional cell culture assay with MTT assay, and we demonstrated that methacrylated version of chitosan is not toxic, and in its toxicology profile, it is similar to unmodified chitosan. In a separate study, we also modified gel and gum by similar chemistry. And this time, we went a bit further. We have conducted in vivo studies when we used this polymer to formulate pilocarpine, anti-glaucoma drug, and we administered these materials, these formulations in rabbits, and we observed what happens with their pupils. Their pupils undergo contraction in the presence of pilocarpine, and we observed the changes in the pupil diameter and confirmed that the formulation with some level of methacrylation provides much better efficiency of the drug effect compared to the conventional gel and gum. But then all these acrylated and methacrylated systems, I still felt that they belong to Professor Bernkop-Schnurch because she pioneered those. And I've been thinking hard, what else could I propose that will kind of be my own suggestion? And then I came across the Maleimide chemistry. Malimides, they are capable of reacting with thiols also under physiological conditions. And our first study we conducted by using a rather complex series of transformations to arrive to a Maleimide containing monomer that was protected. But then we used that monomer to put on a surface of polyvinyl pyrrolidone nanogels, which we synthesized by conventional free radical polymerization. Then we post-polymerized the Maleimide containing monomer on the surface. And then we ended up in the particles that had several cores of PVP, but they were evenly coated with the Maleimide monomer, which was then subsequently deprotected. And then we used it in our studies. So this slide demonstrates an example what happens with protection, deprotection of this monomer using dynamic light scattering. And then we also demonstrated that Maleimide containing polymers, they have much higher mucoadhesive properties, which are comparable with chitosan. In mucoadhesion field, ionic polymers, they are always more mucoadhesive than non-ionic polymers. But in our case, our polymer is still non-ionic, but functionalized with Maleimide groups. It shows the same retention profile as chitosan, which was a good achievement. Later, when I delivered one of the talks in pharmaceutical conferences, I had an interest from several pharmaceutical scientists, and they complained that, oh, your synthesis with malimides is too complex. Can you do something simpler? And then I started looking around, and apparently I found some commercial products available already with malimide groups, phospholipid functionalized with malimides, and we designed liposomes functionalized with malimides, and also demonstrated that these liposomes are much stickier to mucosa compared to their control samples without malimide groups. Another commercial product, a bit more complex, PLGA-PEG block polymers functionalized with malimide groups. We also have conducted a study on making nanoparticles and demonstrated that nanoparticles with malimide, they are showing better mucoadhesive properties. There's several groups also working on malimides, including Professor Bernkop-Schnurch in Israel. They also developed their own system, and some of my ex-students also working in Thailand and recently reported Maleimide-functionalised chitosan. But then a few years ago, I also got inspiration from a work of Professor Justin Haynes from Johns Hopkins University, who demonstrated that if you make materials non-mucoadhesive, if you coat them with low molecular weight PEG, then they will stop being mucoadhesive, but they will develop different properties. They will become mucus-penetrating. And Professor Hayes demonstrated that there's a huge benefit from making the particles more penetrative. In fact, there's still an argument about whether you need to have a mucoadhesive system or mucus-penetrating. So PEGylation was reported, but then I decided to contribute to this field by introducing our systems. So we took a model of Thiolated silica particles. We designed a series of particles which were either modified with PEG, so they were PEGylated, or also we designed systems which were posylated, so they were functionalized with polyoxazolines. And we have used nanoparticle tracking analysis to follow the diffusion of these particles in mucus dispersions and demonstrated that the particles that were modified with PEG and with polyoxazoline, 2-ethyloxazoline at that time, they were more diffusive than the sticky thiolated silica. So this was, our group was the first to demonstrate that polyoxazolines could improve diffusiveness in the mucus. Then I had a fantastic meeting with Richard Hugenboom, and we discussed this study. And I think Richard was the reviewer of this paper at the time. And we agreed that Richard's group would produce some samples of different oxazolines, of different levels of functionalization for us, the same molecular weight, but different levels of substitution with methyl, ethyl, and propyl. And we have conducted another study on comparing these systems, and what we've established that polymethyloxazoline shows the highest enhancement in the diffusivity of these nanoparticles, and N-propyl polyoxazoline is not efficient, is much less efficient, and kind of its efficiency is very comparable to the sticky thiolated silica. Currently, what we're doing, we're trying to, because all these diffusion studies were done with non-porous silica, so we cannot load drugs in them. What we're trying to do currently is to design systems that are porous, that are fully polymeric. So we are currently preparing 2 publications. Both papers are hopefully going to be submitted within a month. In one system, we looked at chitosan that was modified with various polymers. This was also done with, in collaboration with Professor Hugenboom, and we designed nanoparticles with, forgot what is the name of this molecule, tripolyphosphate, with tripolyphosphate. And in another study in collaboration with Dr. Ruslan Mustafin, we've developed a system based on interpolyelectrolyte complexes. When we PEGylated one of the polymers and we designed PEGylated particles, we also demonstrated that these particles show different level of efficiency even in vivo. So to conclude my talk, I hope I was able to demonstrate that there's evolution of mucoadhesive polymers. Currently, this evolution is continuing. There's several groups are working in this field. When we started, it was conventional water-soluble polymers. Many pharmaceutical researchers still use conventional water-soluble polymers in this area. Now there's a lot of pharmaceutical scientists who started looking at polymeric thiomers. There's a number of groups who publish in this area, and there's a few groups working with acrylated and methacrylated polymers and Maleimide polymers. And I hope to convince people that, well, please do work with acrylated and methacrylated and Maleimide-functionalised groups, because this series of polymers, they are relatively unexplored for drug delivery. And also another trend is the mucus-penetrating systems. This trend is mostly predominantly people working with PEGylation, but we demonstrated that polyoxazolines are good for enhancing penetration. Also, there's another paper which we are going to submit soon where we demonstrated some other polymers that could be used. And finally, I need to thank many people. Well, there's a big summary of people I need to thank. I will not be reading their names, but I've been collaborating. Well, obviously, that's a picture of my group, current group, and I've been collaborating with a number of researchers whose names are listed on the slide. I continuously try to update this slide, but it's almost impossible to add everyone. Thank you very much.
Thank you, Vitaly, for being on time. We have actually 2 minutes before the beginning of the last talk. All right, so we have a question from Mike. Let me see. Could you please clarify why do you use PVP particle in the Maleimide polymer particle approach rather than a copolymer particle?
Again, initially we thought about something simple and something established. PVP is a very established polymer in pharmacy. So